Today, our point is cardiac muscles. So, histologically, the cardiac muscle is also helpful for the contraction to all the contraction of the heart. When in resting position, it also state partial contraction or tonus. Histologically, the cardiac muscle also composed of near about 80% of protein. Carbohydrate is present in the form of the glycogen. Lipid is also present. Vitamins, amino acids also occur in the muscles. Inorganic substance present in the form of potassium, sodium, magnesium, calcium, ferrous, copper, chloride, sulfur. All these substances are present in the cardiac tissue also. They are useful in osmotic and acid base equilibrium. So, the muscle is the muscle which is only and only present in the wall of the heart. Hence, it is known as cardiac muscle. This is the tissue of which the heart walls are composed. It is also termed myocardium. It shows certain similarities with both smooth and striated muscles. The cardiac muscle has traditionally been regarded as a branching system. Electron microscope shows it consists of a network of elongated cells of peculiarly striated nature. The cardiac muscle fibers are arranged in layers that form overlapping spirals. The cardiac muscle fibers are about 80 micron in length and 15 micron millimeter in thickness. They are joined end to end and show branching. Where the cell branches join, a bridge is said to have formed. The fibers are separated by strands of loose connective tissue or endomysium through which blood vessels and lymph capillaries and nerve fibers pass. The sarcolemma of cardiac muscle cells is similar but more delicate than that of the skeletal muscle. Each muscle cell contains large oval nucleus which, like that of smooth muscle, is present centrally. The sarcoplasm is quite abundant and contains large mitochondria, a well-developed sarcoplasmic reticulum, Golgi complex, and glycogen. In the atria, there are small granules near the nucleus, which is not seen in the ventricles. These granules have an endocrine function, regulating the excretion of sodium by the kidneys. The myofibrils show cross markings similar to those in the striated fibers. They are absent around the nuclei. In old animals, granules of the pigment lipofuscin accumulate near the nucleus. The most characteristic feature of this muscle is the transverse intercalated discs are present. Each of these discs extend completely across the ends of the fibers. The disc may pass straight across or may show a stair-step pattern. They are formed by interdigitating plasma membrane of cells which are closely linked together by junctional structures. Mature cardiac muscle cells are incapable of division. Under the endocardium, which lines the internal surface of the heart, there is a net of modified cardiac fibers called Purkinje fibers. These form a special impulse conducting system of the heart. Purkinje fibers, as compared to ordinary cardiac muscle fibers, are larger in diameter and contain more sarcoplasm with few myofibrils. They show the central nuclei and intercalated discs. These fibers conduct the stimuli for contraction and coordinate the heart muscle contractions. Contractile action of the cardiac muscle and of heart is involuntary, moderately rapid and occur rhythmically, tirelessly and with small pauses between contractions.